Hello, good afternoon to all the participants participating from Sri Lanka, and good evening to our invited speaker and all the members participating from Australia. I warmly welcome Professor Malcolm McLeod from Australian National University as the invited speaker of the 17th Inspire webinar conducted by the Department of Chemistry, University of Peradeniya, Sri Lanka. Thank you so much, Professor McLeod, for accepting our invitation in spite of your busy schedule. I know it's been a very long day now. It's probably around 11 p.m. And so you know that you are the second invited speaker from Australia for our Inspire webinar series. I hope your presentation will be a very fruitful one to all the participants here today. And also, I would like to welcome all the participants, especially students and the staff members for this 17th Inspire webinar. As you know, this is the second webinar for the year 2022. And unlike in the previous year, so we decided to conduct only one Inspire webinar beginning from this year because of the busy schedule of both the staff and the students after we started our in-person activities. So I hope you all would keep that also in your mind. So without taking much time, I would like to invite Dr. Dimanthi Uduvela to introduce our guest speaker to the audience. So have a great time, everyone. Dr. Dimanthi Uduvela, it's over to you.
Thank you, madam. Good evening, everyone. First of all, let me thank today's guest speaker once again for accepting our invitation to share his research experience with us on this Inspire platform. It's a great pleasure for me to introduce today's speaker. He's Associate Professor Malcolm McLeod from the Australian National University, or the ANU. On a personal note, he was my PhD thesis supervisor at the ANU. He has been absolutely supportive to me right throughout from the day I started my application, and he had been a great advisor for my research project and a great mentor. So much so I've learned a lot from him. So thank you very much, Mal, for all that. So anyone who knows him would call him Mal in short rather than Malcolm. So I'd like to refer to him as Mal to avoid any awkwardness. So Mal obtained his BSc Honours at Monash University, Australia, and his PhD from Cambridge, UK. He returned to a postdoctoral position at the Australian National University in Canberra before moving to his first academic position at the University of Sydney in 1998. Then in 2007, he moved with his group to ANU, where he started as a senior lecturer and later promoted to associate professor. His research interests include medicinal chemistry of nicotinic acetylcholine receptors and the development of new chemical and analytical technologies to study drug metabolism with a particular focus on steroidal conjugates, which frequently finds application in the field of sports drug testing. He has over 1,800 citations on his publications with an H-index of 25. Among the awards he has won, I'd like to bring note of the prestigious Churchill Fellowship Award he won recently to visit leading international drug checking services in Austria, Netherlands, and Portugal, and to rapidly improve the analytical chemistry capabilities in Australia. So he helps the World Anti-Doping Agency to catch drug cheats who abuse performance-enhancing drugs such as steroids, while he uses caffeine as his performance-enhancing drug of choice to boost his activities. And I should mention that he cycles to work every day. So today he's going to talk on new analytical technologies to study sulfate metabolites. So without further ado, I'd like to invite Mel to deliver his talk. Over to you, Mel.
Okay, so thank you. Do I come across loud and clear?
Yes, ma'am.
Okay, very good. Look, it's a great honour to be invited to speak today. Of course, I've had a couple of graduates from Peradeniya through my lab, and they have done a wonderful job. So I am enormously grateful to the education that the students receive at your institution. So let me share my screen and just check that this works. This works properly. And okay, so just checking that looks good.
Yes.
Excellent. Okay, so yes, the topic today, the title at the top, tools for the sulfatome, and what we're going to do is we're going to look at a collection of molecules that are sulfate esters or sulfated, and they're important metabolites. So let me tell you why they're important. I've got an example from steroids here, but it covers all sorts of different classes of compounds. A steroid like testosterone, which of course is present in all of us at various levels, gets metabolized in the body, and it undergoes two phases of metabolism. Phase 1 metabolism, which involves oxidation and reduction reactions to generate a range of different phase 1 metabolites, and then phase 2 metabolism, which involves the conjugation, either with the sulfate ester here or a glucuronide. And so these are the major forms that are excreted in the body, and the reason for that is that it makes them more water soluble. These hydrophobic molecules more water soluble. For the most part, people have studied these glucuronides, and that's because they're probably the major class of steroid metabolite anyway. But there's a close second, which are these sulfated species, and these have largely been neglected. I'm going to tell you about what we've done to sort of rectify that problem. So the first thing we'll talk about is what I'll call indirect detection. And so this is developing an analytical method to target these sulfated metabolites by hydrolyzing the sulfate ester in some way to liberate the free steroid. And this can be very important for techniques, for example, like gas chromatography, where the sulfate conjugate itself is not volatile enough or stable enough to undergo analysis. But once we hydrolyze the steroid, then this is much better. Now, why is this important? Well, let me give you two examples. One of these is a synthetic steroid called methyl testosterone, and this can be abused by athletes in the Olympics, for example. There's a doping case at the moment, I believe, involving steroidal agents. And of course, when this is administered, it may not be the compound that you detect at the end. And so this compound is a major metabolite of this methyl testosterone. And you can see that there's been a range of metabolic changes, including sulfation. And we can detect this compound for 21 days after we administer methyl testosterone. And this is 2 to 3 times longer than any other metabolite marker. Another example here is testosterone. This gets metabolized to epiandrosterone sulfate. And we can analyze this compound by a technique called gas chromatography carbon isotope ratio mass spectrometry to tell the difference between testosterone that you manufacture in your body and testosterone that's manufactured in the lab. For both of these things, it may be necessary to cleave the sulfate ester back to the free steroid in order for the analysis to work. And that's particularly the case for this second example, gas chromatography. So this is quite a time ago, I guess, we had the idea of looking for an enzyme that might help in the analysis of sulfate esters. And we came across this study from 1977, well before I started my chemistry career. And what the authors did here was do a study with an organism called Pseudomonas aeruginosa, which can live in a variety of environments, but that includes your gut. And they grew the bug in the presence of this sulfate metabolite, lithocholic acid sulfate. And what they observed is as the bug grew, they saw the conversion of the sulfate ester to this lithocholic acid. And so this suggested that the Pseudomonas aeruginosa, the bacterium, had a sulfatase enzyme that it could express to harvest the sulfate from this sulfated metabolite. What's more, if you took the sulfate source away, the bug stopped growing if there were no other sources of sulfur in the environment. So this would seem to be good evidence for a sulfatase mechanism of action. So these are really, sulfatase enzymes are really interesting enzymes. They start when they're first expressed with a cysteine or sometimes a serine in the active site. They get post-translationally modified to this formal glycine residue, which can serve as a catalytic nucleophile to hydrolyze the sulfate ester by cleaving the sulfur-oxygen bond here. You end up with this sulfated intermediate, which can eliminate hydrogen sulfate and then be hydrated back to the active species. So, of course, I know some of you in the audience will know quite a bit about this, but where we started was to do a study, I guess, of this enzyme. So we were able to get the gene, express the gene and purify the enzyme, and then test it for the hydrolysis of steroidal sulfates. And we did that initially against a panel of 8 different sulfated species. And you can see them small in the right-hand side here. And it's worth, don't worry too much about the exact structures or names here. Just realize that there are different stereochemistries and positions of attachments for these sulfates. We also tested not just the Pseudomonas aeruginosa enzyme in red here, but also a number of commercial enzymes. So the one at the top here is Helix pomatia, and it's derived from the Roman snail. And in the table, you can see these numbers, and the bigger the number, the faster the hydrolysis. So 6 is good and 0 is no hydrolysis at all. And you can see if you look at the top 2 rows, that the Pseudomonas aeruginosa enzyme was about as good, comparable to the best commercial enzyme, Helix pomatia. We thought there were some advantages in that the enzyme was purified, so it wouldn't contain other activities. And we also, because we had the enzyme and the gene for the enzyme, we thought we could improve on the enzyme activity by doing some engineering, protein engineering. So what we aimed to do then was to studying this hydrolysis reaction again, testosterone sulfate, hydrolyzing to testosterone. We're going to take the gene for the Pseudomonas aeruginosa sulfatase, I'll call it PAS from now on, and we're going to mutate that gene. So we can mutate that in a variety of different ways around the active site. We can then transform E. coli and grow colonies containing those mutant genes. We can screen those using liquid chromatography mass spectrometry to see which mutants do the hydrolysis the best. We can confirm those hits, and eventually we can purify the enzyme and do further studies. Now, this is not the end of the story, because if we find a mutant that's better, then we can feed it back into the process. It's iterative and go round and round again. And of course, you might recognize the name of the author here. This is Demanti's work, and I just touch on it briefly as a starting point, because we will come back to this a little at the end. But to cut a long story, a very long story short, the research was enormously successful, and we found a range of improved mutants. We found 3 here that were over 150 times better than the starting point, our wild type PAS. And so we're measuring here Vmax on Km, which is just a measure of enzyme efficiency. And the higher the number, the better. We also found some mutants that had broader substrate scope, in addition to being faster at hydrolyzing steroidal sulfates. So this was great. Now, this work has been applied a couple of times now, and I'll just briefly describe those. So this is one of the examples we saw on an early slide. So testosterone is metabolized to epiandrosterone sulfate. And we can use epiandrosterone sulfate as a marker to see whether this testosterone is naturally occurring or is synthetically derived. And the way we do that is to hydrolyze the sulfate. And for that, the lab in question, this is working with Professor Thevis at Cologne, a wider accredited lab. So hydrolyze the sulfate to get epiandrosterone, and then we do this gas chromatography, carbon isotope ratio mass spectrometry. And here's a couple of plots of what's going on here. Let's just look at the bottom one, perhaps. There's just two volunteers. And what this plots is samples from a volunteer, urine samples that we're processing and analysing using this technique. And this orange bar indicates the days at which they're applying a testosterone gel, so an external source of testosterone. So they're doping. And then what we're looking at, these traces here are different markers. So the one at the top is a reference compound. This is something that we don't expect to change, and indeed it doesn't. Then the open diamonds here, or the open markers, are different markers for testosterone. One of them is testosterone itself. And you can see as the drug is administered from day 3 to day 9, you can see that the carbon isotope ratio, which is on the left-hand side, the y-axis here, changes and decreases until you stop treating the person with the testosterone gel, and then it rapidly returns about to where it was originally. Well, what about the black trace? This is now epiandrosterone. And you can see that the black trace also decreases, but interestingly, it stays depressed after you cease the treatment with testosterone. And so the idea of this marker is that we can detect for a little longer how the testosterone administration by monitoring for this epiandrosterone species. There's been a recent application of these enzymes as well. Again, I'll just briefly talk about this one, vitamin D. And so vitamin D, obviously it's a critical hormone, important for bone development and a range of other functions. And of course, we get our vitamin D from a really interesting series of reactions, from dehydrocholesterol, the action of UV light. It undergoes an electrocyclic ring opening and a series of other conversions to eventually give this precursor to the vitamin, 25-hydroxy vitamin D. This is one step away from the active hormone. Now, it's common for medical scientists to analyse for this compound when they're trying to assess the level of vitamin D in a person. And so we worked with a group in the Anzac Institute, Carl Jenkinson and his colleagues in the Anzac Institute in Sydney, to look at vitamin D status and see what effect conjugation of the hydroxyl group had on the vitamin D. And so the first experiment here is just analysing vitamin D. There are 3 groups in all of these plots. One is just just a normal population, and the second 2 are supplemented with either vitamin D3 or vitamin D2. And you can see when you analyse for this 25-hydroxy vitamin D3, you get between 20 and 30 nanograms per mil of vitamin D for some of these, and a little less in the D2 supplemented group. Well, what happens if we apply the hydrolysis enzymes? So we're going to use glucuronidase and sulfatase. And now what we see is that the level of these different groups double roughly. What this means is that roughly 50% of the vitamin D precursor here that's circulating is conjugated either as the sulfate or the glucuronide. And what's more, if you look at these plots down below, then the one on the left, this is the sulfate form, and you can see this is higher than the plot on the right, which is the glucuronide form. So what does this mean for analysing vitamin D levels? Well, it means that instead of just analysing for 25-hydroxy vitamin D3, which is roughly half of the vitamin D, this form of vitamin D that's circulating in your serum. You should also look perhaps for the glucuronide, although this is rather minor, just 4%, but certainly for the sulfate form, shown on the right-hand side here. And of course, in order to assess the vitamin D status, perhaps what we should be doing is hydrolyzing these different forms. So using one of the mutant Cas enzymes that was developed in my lab, or E. coli glucuronidase, you can convert these conjugated forms to the free form and analyze the combined vitamin D fraction. Okay, so that was a brief introduction, I guess, to one area of our research, looking at engineering enzymes and sulfatase hydrolysis. And just the conclusion is that we have this Cas enzyme, and it can hydrolyze a range of sulfate metabolites. We've seen steroids and vitamin D, and I'm sure there'll be more applications in the future. Furthermore, we can engineer these mutants for specific activities. And so we've done that to increase activity, but also substrate scope. So I thought I'd move on now to a slightly different topic, which is, again, related to these sulfate esters, but direct detection. So wouldn't it be easier if, instead of having to hydrolyze the sulfates prior to our analysis, if we could just detect the sulfates directly? And so this is a little bit of a discussion about how we might do this, and also focusing on some rather unusual sulfate metabolites. So this work started with a sabbatical in Barcelona some time back, and the question that we asked at the start of this sabbatical involved the metabolism of methyl testosterone and the generation of this metabolite here. And it was interesting, this transformation was rather interesting, because if you look carefully, you can see that the stereochemistry at the 17 position here on the 5-membered ring has changed. And we wondered how that might have occurred. And so the proposal was that this methyl testosterone underwent phase 1 metabolism to this diol complex, then perhaps sulfation from the doubly sulfated species, and then this tertiary sulfate was unstable and would decompose and hydrolyze with an inversion of configuration to generate the metabolite that's observed. Okay. So this seemed like a reasonable proposal, but the question, it raised a question, an important question, as to whether these sort of doubly sulfated species actually existed, whether they were real or not. And so we decided to answer this question by a process of synthesis first and then analysis. So it's relatively easy to make these doubly sulfated compounds. You just take a steroid or diol, you can react that with a sulfur trioxide amine complex of some type, dimethandioxane. After a while, the sulfation is complete. And we use solid phase extraction with weak anion exchange resin to purify the sulfated species away from all the other reaction components. Now, the conversion here is very good on a small scale, and the yield is high on a larger scale. And it says 12 examples, but I'm sure we've done many, many more than this now. So now that you've made a collection of these doubly sulfated compounds, now you can study their mass spectrometry. And so the first thing we looked at was how these compounds ionize. And as you might expect with these sulfate esters, they're both very acidic groups. We tend to see the dianion as the predominant species, right? And only a little bit of the monoanion. So you can see the mass of this dianion is 450 Dalton, but because there's 2 charges, the mass to charge ratio is 225, or half of that. We can then study the fragmentation of this dianion in a triple quadrupole mass spectrometer by a process called collision-induced dissociation. So here we accelerate the ion, we smash it into a collision gas, argon and nitrogen typically, and we look at the fragments that occur. And what we see for these types of compounds typically is loss of hydrogen sulfate, syn elimination and of hydrogen sulfate to generate an alkene in this monosulfate fragment. And so you can see this, this monosulfate now has only one charge, 353 Dalton, or mass to charge ratio 353. So it's a rather interesting association in that the mass to charge ratio actually increases by M on Z 128. Normally fragmentation leads to smaller fragments, but we've changed from a doubly charged to a singly charged fragment, so it goes up. What's more, we can write out a mathematical formula that explains that change in mass to charge ratio from the precursor to the product. And for the case shown here, the formula is rather simple. It's the increase in mass to charge ratio is simply the mass to charge ratio of the precursor ion minus 97, which is the mass to charge ratio of the fragment, the sulfate fragment. And with this little formula, you can derive a table of maybe hundreds of different transitions that relate these precursor ions to the product ions. And if we look for these transitions in a mass spectrometer, then we can analyze for a wide range of these different doubly sulfated species. So I'll just give a couple of examples. So we call this process constant ion loss because it starts with a range of different precursors, but it always involves the loss of a particular sulfate fragment. So let's show a couple of examples of using this technology in analytical chemistry. And I'll start with another doping-related study. Again, this was conducted with collaborators in Barcelona. And this is the steroid tibolone, which again is prohibited by the World Anti-Doping Agency. And if it's given to athletes, then one of the metabolites that's generated is this compound here. There's been some reduction and a twofold sulfation to generate this bisulfate. And once again, we can do our direct detection of this bisulfate here using our ion loss strategy. So taking this precursor, colliding, and looking for the product ion here, we can detect this molecule in athlete samples. So here's a urine sample. This is before we administer the drug. And again, we're looking for this transition. And you see that this is a fairly flat baseline. Here's a sample urine sample post-administration of this drug tibolone. And we can clearly see the transition here for this doubly sulfated metabolite. So we're using this ion loss chemistry, mass spectrometry, to detect these types of steroid metabolites associated with doping. Now, this is not where the story ends, because there's been a more recent application of this technology, and this is in prenatal diagnosis. So this is diagnosing disease in unborn babies. So when the baby is in the womb, it begins at some stage. It begins its steroid metabolism, and this is, of course, to generate the steroids it needs as hormones and other things. And so you can see the pathway here down through from dehydrocholesterol down to cholesterol and all the way through to other steroids like DHEA and so on. So this begins in the unborn baby, and the steroid metabolites are exported through the placenta, which can also undertake some metabolism. So now we're forming estriol. And finally, it's excreted in the maternal urine as estriol glucuronide, and here's the structure on the right-hand side. Now, there are some conditions of steroid biosynthesis where there are deficiencies in an enzyme called P450 oxidoreductase. And if we block this P450 oxidoreductase, we get a condition called PORD, or P450 oxidoreductase deficiency. And this blocks a number of these metabolic transformations. And so we get less of this estriol glucuronide. We also get a buildup of the precursors, which end up being excreted through the placenta as this pregnine derivative. and then in the maternal urine as a pregnane derivative and a bisulfate, doubly sulfated. So again, using this ion loss analytical process and then synthesizing and confirming these compounds, we can identify the bisulfates that are markers of this blockage in steroid biosynthesis. So just one result again. So here's a normal urine sample, a normal pregnancy, no deficiency, and we can see here's estriol. This chromatogram here shows estriol, 16-glucuronide. This normal end product here, quite a large peak, and only a small peak on the slightly to the right here for this pregnane bisulfate. But when we have a case that's positive for this deficiency, then we see a dramatically different situation. So the estriol glucuronide diminishes significantly, almost disappears, and we get much, much more of this pregnane diol bisulfate. It's very good to take a ratio of these two peak areas because that leads to a very nice marker for detecting the presence of this deficiency in unborn babies. I'm going to skip tack here to looking at introducing labelled, stable isotope labelled steroid sulfates. So we can make sulfates, but we can also do something else, which is make stable isotope labelled sulfate derivatives. And so here's testosterone sulfate with 3 O18 atoms as part of the sulfate ester. We can make this fairly straightforwardly using stable isotope labelled sulfuric acid. And this has a high level of O18 incorporation. We dehydrate this by reacting with acetic anhydride in pyridine and then react it in the normal way to form the sulfate ester. So we did many examples, again, of this labelling. The conversions are generally quite good. And you can see on the right-hand side, here's the mass spectrum for this labelled compound. Now, this is epiandrosterone sulfate, another steroid sulfate we've met before. And normally, this has a mass to charge ratio of 369. But you can see here for this synthesized labelled derivative, we can see we have a big peak for the +6 peak, 375, smaller peaks for +4 and +2, and none of the unlabelled derivative. And what this means is that we can use this compound, this isotope labelled compound, as an internal standard. And this is really useful for quantifying compounds because you can take the ratio of the unlabelled analyte to the internal, the labelled internal standard. And this is a very common way that quantitative analysis is undertaken. Just a little comment on the cost of this process. The labelled sulfuric acid is very expensive, over $1,000 US a gram. But the process is not too expensive when you think about a steroid derivative. Making 5 milligrams, which is a lot for mass spectrometry, can last for a long time, only $23 a reaction. So we can do many, many reactions with this gram of labelled sulfuric acid. Just the last thing I should have mentioned. Of course, we can observe ratios for the +6, +4, +2 and unlabelled species in our labelled derivative. And we can also compare it to theory, the theoretical extent of labelling, and this matches up more or less fairly well. So a couple of other examples of this stable isotope labelling process, and this is making these doubly sulfated steroid derivatives. So we're going to start with estrone sulfate here. We can reduce estrone sulfate to the alcohol and then sulfate with our labelled derivative. So this selectively introduces the second sulfate up on this 5-membered ring of the estrone bisulfate molecule. Same sort of thing for this 16 hydroxylated derivative. Now we're starting from a doubly sulfated precursor. We're going to use one of our mutant enzymes to cleave off the 16 sulfate selectively and liberate this alcohol. And then we can introduce the stable isotope labels selectively again at the 16 position. And what's good about this is that in the past, when we've made these bisulfate molecules, the sulfate units at each end have been the same. And so we haven't really been able to study what happens when each end undergoes some sort of change. So the first thing we looked at here was, of course, the fragmentation. And this is a product ion scan, another mass spectrum of this molecule here, estradiol bisulfate, selectively labeled at the 17th position. And you can see the precursor here, the dianion here, occurs at 218 mass to charge. To the right-hand side, after we fragment, to the right-hand side, we can see some steroid monosulfate fragments. So, for example, in black here, this is the monosulfate fragment where we've lost labeled sulfate, and in red, where we've lost sulfur trioxide radical anion from the phenolic sulfate to generate this radical anion species. So you can see these two peaks to the right. And of course, to the left, we can see a number of sulfate-derived fragments that correspond to those fragmentations, and also this doubly charged fragment at 178 mass to charge ratio, where we've lost sulfur trioxide as a neutral loss. I think that's a bit hard to see looking at that spectrum, what's going on here. So I've drawn it out in a kind of tree-like diagram where we can look at the different fragmentation processes. And so for estradiol bisulfate, we actually see three. We've described them already. Loss of hydrogen sulfate and generation of this alkene-containing monosulfate fragment. We see loss of sulfur trioxide radical anion and generation of this radical anion, and then in the middle, formation of this dianion through neutral loss of sulfur trioxide. We can go even further because we can modify the mass spec conditions, modify the ionization that can generate some of these fragments in the source, and then we can select them again and fragment them further. So we can see how these individual monosulfate fragments fragment further on further collision-induced dissociation. And we can start to unpick some interesting features of these different molecules, which may allow us to identify them later on in analytical chemistry. So just to conclude this section, we've done a synthesis of some steroid sulfates and some steroid bisulfate compounds. And I think this is relatively easy. And if you ever need these for your research, go ahead and give it a try. I'm sure you'll succeed. Of course, we use these as what we call reference materials or standards. And these enable us to do new analytical chemistry. They've enabled us to do develop direct detection methods for these rather unusual doubly sulfated species. And we've applied this to doping and prenatal diagnosis. We've also done synthesis to introduce stable isotope labels. So these are these O18 labels into our bisulfate derivatives. And we can do so selectively and then use these in mass spectrometry, either as internal standards or as probes, I guess, to understand the fragmentation of these types of species. So I have one more topic, which is going even further, I guess. So we started with indirect detection and hydrolysis of steroid sulfates, then direct detection of particular sulfate classes. Now what we're going to talk about is the direct and untargeted detection of sulfate metabolites. And so the aim for this section is to develop a method that can detect any sulfate metabolite in principle and characterize its behavior in various ways. And so I'll show you a couple of applications. So the basic idea here is to use, now we're using, instead of a triple quadrupole instrument, we're using a high resolution mass spectrometer, and we're doing tandem mass spectrometry. So the idea is to use liquid chromatography, high-resolution tandem mass spectrometry with our samples, controls, and what we call pool QCs to gather some data. What we're then going to do is we're going to take the normal scan mass spectrum. We're going to take that data and we're going to do something called data-dependent acquisition, and we're going to align fragmentation data to the original scan data. So in essence, we're going to fragment our molecules and we're going to look for characteristic sulfate fragments, and where they occur, we're going to annotate our list of metabolites with this information. And then using this, we can do a couple of things. We can classify things as sulfates or non-sulfates. We can look at their levels and how significant they change. And then hopefully we can begin to identify this collection of molecules and use that information. So the first study I'm going to give you today is another doping study, but this time it's testosterone propionate. It's a common veterinary drug, and we're going to administer this to a horse. And of course, horse racing in Australia is big business. Many millions of dollars are wagered on horse racing every year. And so there are labs that are working to ensure that the industry is clean and free of cheats, I guess. There's also an important aspect of animal welfare. So if animals are being treated with inappropriate drugs at inappropriate times, then it can harm their health. So again, what we're going to do is we're going to study, we're going to administer the drug to the horse. We're going to study the sulfate metabolites that come out, and not just one metabolite, but as many as we can see in an untargeted approach. So the study design was to take some equine urine for a single horse before administration, the day before, and 12 hours after administration of the drug. We're going to prepare our samples in triplicate along with controls and pooled QC samples, which I'll explain in a little while. Sample preparation is very simple. We just use solid phase extraction, centrifugation and solid phase extraction to generate our extracts. And then we do liquid chromatography with an acetonitrile water gradient on a phenylhexyl column, and it's a 30-minute runtime per injection. So the mass spectrometry we're going to do is a bit more complicated. So it has two different types of scanning. The first thing we do is what we call a full scan or a scan mass spectrum. And this is just scanning for all of the ions that we detect at a certain time point. Next, what we do is called data-dependent acquisition. And so we pick the major ions that we see in this scanned mass spectrum, and we select them and we fragment them, and we collect a second tandem MSMS spectrum, which has information on fragments. And we do that 2, 3, 4, 5, maybe 10 times for each scan window. So we collect information about the fragments for the major ions. Now, these two scans are done at different resolutions. So 70,000 for the full scan and 17,500 for the data-dependent MSMS scan. We also use what's called static and dynamic exclusion lists. So a dynamic exclusion list just means that if we select an ion here in this scan window, then we won't select it for the next 3 scan windows. And this means that other ions can be analysed. The other thing we do is what's called a static exclusion list. And so this is where we look at our solvents and our extraction protocols, and we find all of the ions that arise from those basic processes, but don't arise from our sample, and we exclude those as well for the duration of the run. So I'll just say a little bit about pool QCs and alignment and quality control. So the idea of pool QCs is that we might have a series of samples over on the left here. These might be drug-treated samples or control samples, and we prepare those in triplicate. And then before we analyze them, what we do is we take a little bit, a small part of each of these samples, every one, and we combine them together and mix them up, and then aliquot them out into a number of what we call pooled QCs. What this means is that every metabolite that appears in these samples should be represented in the pooled QC, which is then aliquoted out and analysed periodically through our analytical measurements. So, of course, we have a number of samples to inject, and we have a number of QCs as well. What this might look like is something like this. And so we have a range of samples with a range of results, and you can see in red the pooled QCs. And we can use this in two ways. So we can use the pooled QCs to help us align the data. So sometimes retention times can drift a little bit in liquid chromatography mass spectrometry. But if we have pooled QCs throughout, then we can use these to align all of the data together. So the same peak appears at the same retention time for every sample. The other thing we can do, you can see that these pooled QCs are a bit wavy. They go up and down, and that's not uncommon for mass spectrometers. Their response can vary. But because the pooled QCs are all the same, we can then normalize the data. And so we can account for this variation. Now, the last thing pooled QCs allow us to do is a bit of quality control. So we can check that our data is good. And so the way we've done that is to take all of our data and do what's called a principal component analysis. So this just takes our different samples and it looks to see how different they all are to each other. And this is a three-dimensional principal component analysis. You can see three axes here, and it's a box. And all of our controls and samples and pooled QCs appear in this box. So in blue, light blue in the middle here, are our pooled QCs. And it's good to see them in the middle because, again, they're a representative sample from all of the different species that are present here in the analysis. Over on the right-hand side here in sort of brown compound, this brown colour, this is our reddish colour. This is our plus 12-hour horse. This is after drug administration. Here's before drug administration. And here's a control horse, which we've analysed at 3 separate time points. And you can see they're all rather similar. So this is good indication that our analysis is working well. So, okay, we've taken our drug, we've given it to a horse, we've analysed the urine. What do we see? Well, once we've done our analysis with our sample, our controls, our pooled QCs, we align and we normalize the data. What we get is 6,230 different features there. When we add the MSMS data for sulfate reporter ions, this comes down to 3,462 features. Some of our features here don't contain this data-dependent MSMS data. And then we can use some algorithms to cluster the data into sulfates and non-sulfates and to look at the levels. And now we get down to 962 sulfates. Let me just take you through this process in a little bit of detail. Let's have a look at sulfate fragmentation. It's very common for sulfates here to fragment, and typically they fragment to these 6 different species with all different mass to charge ratios, or these 2 at the ends are neutral losses, sulfur trioxide or sulfuric acid. The other thing about these sulfate metabolites is that when they fragment, they typically give sulfate fragments as the major fragment and few other ions. Okay, so we have a small collection of sulfate-derived fragments, and they tend to be the major species. And so we can use this information from the MSMS spectra to classify all of the features, the 3,467 features, into sulfates in yellow here, or non-sulfates in black. And you can see that the sulfates in yellow, these on the y-axis here, is the maximum abundance. So this is reporting the maximum abundance of the sulfate reporters. And you can see there's a lot of them up at the 100% mark. as opposed to the non-sulfates in black, where we see a few sulfate reporters and typically not of high abundance. The other measure we use is intensity ratio. And again, when we have sulfates, they tend to be the major fragments that we observe. And again, we see that in the yellow section here, whereas for the non-sulfates clustered, we see lower proportion, I guess, of sulfate fragments. So we've used an algorithm here called k-means clustering to split the data into two groups, sulfated and non-sulfated. Here's just some violin plots. So you can see in here, this gives an idea for where the mean of, for example, the maximum abundance or the intensity ratio is. And you can see that these are really well separated. So we've successfully separated sulfate from non-sulfate. We can do a few other things. We can look at speciation. So we said we had 6 different sulfate-derived fragments. And so we can look at our data and see which of these appear as the major fragment. And you can see that we have, sulfur trioxide radical anion is roughly half of our sulfate metabolites fragment to give this fragment, followed by sulfur trioxide, neutral loss, followed by hydrogen sulfate, about 20%. And we know that these are associated with different classes of compounds. So, for example, saturated sulfates tend to give hydrogen sulfate, a little bit of this sulfur trioxide radical anion. Estrone sulfate tends to give neutral loss of sulfur trioxide, plus again, some of this radical anion. So this tells us something about the types of sulfates that we're seeing in our equine urine. Of course, we're doing a doping study, so we want to find compounds that are upregulated after doping. And so here's what's called a volcano plot. On the y-axis is just a log fold change. So if it's a positive number, it means that the metabolite is increasing in concentration. And on the y-axis, we just have a log to the base 10 of something called the adjusted p-value. So this is a measure of significance, I guess. And we've used a p-value of 0.01. So the grey here is non-significant, and if it's blue or red, then it's significant. And of course, for this doping study, we're interested in the molecules that appear in this top right corner. So after testosterone propionate administration, we get metabolites that are upregulated and highly significant. So you can see these little dots up here. And in fact, this worked very well. We were able to identify 10 metabolites of testosterone propionate that were upregulated significantly, and you can see the fold changes here. 4 of these were confirmed against reference materials that we synthesized in the lab. One of these was testosterone propionate. We've got this androstenediol, epiandrosterone sulfate, which we've seen before, and this diol monosulfate, which was a new metabolite for equine analysis. There are a number of other metabolites down below here that remain unidentified, and largely they remain unidentified because they involve additional transformations like hydroxylation. And the number of combinations, I guess, that we would have to investigate in order to identify these is quite large. So we've left that for the time being. But we might come back to these if they turn out to be particularly interesting in future studies. So we do have more work to do. We have to do studies over time, for example, in individual horses, but also studies of populations of horses to make sure that these are indeed markers of testosterone administration and not just don't just change with normal variation. So there's some work ahead of us. The second story I want to tell briefly is again looking at enzyme hydrolysis, and it harks back to the beginning of the talk and using enzymes to cleave sulfate esters. And we're going to use the same methods now to investigate all of the sulfate metabolites as they are hydrolyzed by a collection of esters. So for this study, we used a pooled urine sample. We collected samples from 3 males and 3 females and mixed them together. We either treated them with no enzyme or with our PAS enzyme, the wild type and 2 mutants, or with the commercially available helix sulfatase enzyme. So the sample preparation is quite straightforward again, similar to the last time, and similarly the chromatography, this time a methanol water gradient and a C18 column, so slightly different conditions, but the same basic process. Again, with all of our samples, controls and QCs, we run a lot of samples and we get a lot of data. We align these and look at the features. There are about 15,000 features. Of those, only about 5,700 contain MSMS information, for example, of sulfate reporter ions. So these are the ones we analyse further. And of these, around about 1,430 turned out to be sulfate molecules. Okay, so again, here's our volcano plots. And again, on the x-axis here is log fold change. And now we're interested in this negative region of the plot, because a negative log fold change means that the metabolite decreases. So if we hydrolyze a sulfate, we should see that level of that sulfate decrease. And you can see here's all features, so sulfates and non-sulfates on the left-hand side. On the right hand are the sulfated features, and you can see this region here. These are strongly downregulated, consistent with enzyme hydrolysis. Again, we can look at sulfate speciation, a similar plot to what we've seen before. It's a little bit different this time. So in the horse urine or the equine urine, we saw this sort of distribution of these different ions, different fragment ions in our sulfate metabolites. And in the human samples, it's changed a little. And so we're getting slightly less of this sulfur trioxide radical anion. And this suggests to us that we've got fewer unsaturated sulfates in human urine. Whereas we get more of this hydrogen sulfate. So this indicates to us that we've roughly doubled the number of saturated sulfates in human urine when compared to horse urine. So some interesting trends there. We can also look at the speciation of the fragment ions for the sulfates that are hydrolyzed. So this is quite interesting. So what we observe, so this plot here has, for each of these different reporter ions, has 5 bars. The first one here is what we see for all sulfates. And the next 4 are what we see for the different enzyme treatments and the sulfates that are hydrolyzed. And you can see that this M on Z 80, sulfur trioxide radical anion, is underrepresented, whereas 80 Daltons, the neutral loss of sulfur trioxide, is overrepresented. And what this suggests to us is that hydrolysis by all of these enzymes tends to favour electron deficient and unsaturated sulfate esters. So just to explain that, unsaturated sulfate esters, we think, fragment in this manner. We get bond homolysis to generate sulfur trioxide radical anion and this phenolic radical. But then we can also get an electron transfer back to the phenolic radical, so a reduction to get this anion. And now this is the neutral loss of sulfur trioxide that we're observing. And of course, electron deficient substrates would tend to favour this electron transfer back to the steroid radical or the radical fragment. And in fact, for estradiol sulfate here, this is the major pathway. Now, of course, for enzyme hydrolysis, why does this matter? Well, electron withdrawing unsaturated sulfates have better leaving groups. So this phenolate anion will be stabilized when it's hydrolyzed by the enzyme. So we think that's a marker, I guess. Speciation here is a marker of the selectivity of the enzymes for these unsaturated sulfate esters. Interestingly enough, looking at our PAS enzymes, we're looking over on the right-hand side, we maybe see a small increase in the ability of our enzymes to hydrolyze saturated sulfates that give rise to this MONZ97, or hydrogen sulfate. And of course, this was how we engineered our enzyme to be able to hydrolyze saturated sulfates like testosterone sulfate. So maybe this is understandable. So lastly, let's look at the overall results for hydrolysis of the sulfates in equine urine. So that's this plot here, and there are three regions. They're all sulfates. Down the bottom, we have sulfates that don't change. In the middle, the red arrow here, we have sulfates with a negative fold change consistent with hydrolysis. And at the top, we have sulfates with a positive fold change. And what we can see for this negative fold change consistent with hydrolysis, all of the enzymes hydrolyze about 27% of the sulfated metabolites. It means that enzyme hydrolysis is not a perfect tool for analyzing sulfate esters. But there's some other interesting things that we can see here as well. So, for example, these are different enzymes, of course, and Helix Pomasha here has this rather large box here, which is a positive fold change. This means that the sulfates increase in concentration, and this doesn't seem consistent with sulfatase activity, right? Enzyme hydrolysis, a little unusual. And if we look at the non-sulfated features here, again the same enzymes, looking at the same sorts of things, no change, negative fold change, and then positive fold change. Again, what we see is Helix Pomasha has a large fraction of positive and negative change for the non-sulfate fraction. And this is not what you'd expect for sulfatase enzyme hydrolysis. And what this tells us is that Helix Pomasha sulfatase is a crude enzyme preparation, and it's very poorly selective and has many alternative activities that we can see in this right-hand plot here. So going right back to near the start here, we had this table where we said, okay, Pseudomonas aeruginosa sulfatase looks about the same as Helix Pomasha. But now I think our conclusion can be stronger, and that is that the Pseudomonas aeruginosa sulfatase enzymes are much better because they don't have alternative alternative enzyme activities, and they lead to a selective hydrolysis of the sulfate fraction, or more selective. So just to wrap up, again, this is a section on untargeted direct detection of sulfate metabolites, and the nature of this screen is much more comprehensive than what we've done before. And of course, we've used this in the doping sense to identify four metabolites of testosterone propionate in horses. And then we've used the same system to study enzyme hydrolysis between the PAS enzymes and Helix Pomasha. And the strong conclusion is that we should use the PAS enzymes, wild-type PAS or its engineered variants, if we want a selective analysis of the sulfate fraction, noting, of course, that only a fraction of sulfates will likely be hydrolyzed. So I need to thank a lot of people. Firstly, from the School of Chemistry. So the key people who did the work, of course, Demanthi originally did the enzyme engineering study, and Chris Fitzgerald, who did the bulk of the work on steroid, on the sulfate labeling and the untargeted screen. There are a range of other contributors to this chemistry who I thank enormously, from both my group, but also from the analytical services of the ANU. I'd also like to thank my collaborators around the world, Oscar, Thomas, Cedric, Lance, and Carl, for their assistance. And I've shown some of their applications of our research in collaborations throughout the talk. And finally, I'll just leave you with a picture of Canberra. This is sort of midwinter in the Balloon Festival, which occurs at dawn. Very nice. And lastly, our new building of Research School of Chemistry. Now, I don't know, 5 or 10 years old, I guess, but still in very good nick and an excellent place to do research. So I'll leave it there and ask if there are any questions.
So thank you, Professor McLeod, for a very interesting and detailed presentation. So I'd like to invite any questions from the audience. Either you can unmute and just ask it directly, or you can type in the chat. And if you're asking through chat, please type to everyone so Professor McLeod can also see the questions. Professor McLeod, I actually have a very general question.
I consider it a great honor to propose the vote of thanks this evening on behalf of the Department of Chemistry, University of Peradeniya, and members of the INSPIRE team. Firstly, I would like to express my sincere gratitude to Professor McLeod for his insightful talk on new technologies to analyze sulfatomes. All of us gained remarkable knowledge on new analytical techniques which can be used to study sulfate metabolites. So thank you, sir, for taking time out of your busy schedule to share with us an evening full of incredible scientific knowledge. Next, I would like to extend my heartfelt gratitude to Dr. Dimanthi Udwala for inviting and hosting the guest speaker today. Thank you, madam, for your constant support and motivation. I would also like to thank Professor Manav-Devi Ganahenage, Head of the Department of Chemistry, for her guidance. Madam, we are always grateful for your support and encouragement to make INSPIRE a success. At last but not least, I would like to thank everyone who joined us today to be a part of yet another wonderful INSPIRE session, and hope you found the evening valuable as much as we did. Before winding up today's session, I would like to invite you all to our next webinar, which will be held on 10th March 2022, and the invited speaker is Professor Wolfgang Fritzsche from Leibniz Institute of Photonium Technology, Germany, and he'll be speaking on bioanalytics using plasmonic nanostructures. Thank you, everyone. Have a pleasant evening.